I think at this point it's reasonable to ask the following question, which is, why are we spending so much time on this? Why is chirality, or why are stereoisomers are important? The first reason is that, here are two stereoisomers of each other. They really look alike. At first glance, you wouldn't necessarily think that those things are different from each other. But they really are different from each other. They're not the same thing. So, first of all, looks can be deceiving. That's one answer. It's not super satisfying. I think the next answer is probably more satisfying. Many, if not most, biological molecules are chiral. In other words, many or most biological molecules come in left-handed and right-handed or mirror image forms. So again, maybe you might say, who cares? Here's an example of an answer to who cares. This molecule here is called D-glucose. Those of you who are familiar with glucose, glucose is a pretty common type of sugar that sometimes is given intravenously, but it's a sugar that is used a lot by us. We get energy from it. This is the arrangement of atoms. This is the structure of a molecule called D-glucose. It's a type of sugar. And just to help you navigate each of these corners and intersections, so over here there's a carbon atom, carbon, 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 and there's a carbon here as well. And there are different atoms attached to those carbons, but that's just to help you orient yourself. That's D-glucose. D-glucose has a mirror image molecule that is not identical to itself. This is L-glucose. It has obviously a slightly different name. You don't need to know what the D and the L stand for, but hopefully you can see that they are mirror images of each other. If you put a mirror down the middle here, the reflection of D-glucose would make L-glucose, and vice versa. If you put a mirror down the middle, the reflection of L-glucose would make D-glucose. These molecules, even though they are very similar looking to each other, they're not the same thing. The one on the left tastes sweet to us, and we can eat it, and we can get calories from it. We can use it for food. We can get energy from this thing. The molecule here on the right, L-glucose, it tastes sweet as well, but we actually can't get any energy from it. Our bodies cannot use this thing for food. The punchline here, and I know everybody likes that phrase, punchline, the punchline here is that your body can actually tell the difference between left-handed and right-handed molecules. So your body is clever enough, sophisticated enough, that it can tell the difference between this thing on the left, that molecule, and the molecule on the right. So much so that it can basically use this and break it down and get energy from it. And if you ingest this stuff, your body basically looks at that and says, I have no idea how to get energy from that, and it just passes through your body. The reason that that is true, the reason that your body can tell the difference between many left-handed and right-handed molecules, is because many of the biological molecules in your body are also left-handed, either left-handed or right-handed. And it just so happens that the molecules that are used to break down glucose, to get energy from glucose, have either a left-handed or a right-handed form that will only attach to this type of molecule and break it down and get energy from it. And those molecules in your body that are responsible for breaking down glucose, they have no idea how to attach to this particular molecule here. So I mentioned that you can think of these things as left-handed and right-handed molecules. The way that I want you to think about this right now is I want you to think of this molecule as being like a left hand, and this molecule over here as being like a right hand. And the molecules in your body that are responsible for recognizing sugars like glucose and breaking them down for energy are like a left-handed glove. And only the left-hand molecule is going to fit inside of the left-handed glove to be broken down. This is like a right hand trying to fit inside of a left-handed glove in your body. And because it doesn't fit properly, the molecules in your body that are responsible for breaking down glucose and using it for energy can never properly attach to this molecule and they can't break it down. So the punchline is that a lot of your biological molecules that are responsible for doing important things in your body very often only come either as a left-handed form or a right-handed form, and not both. And because of that, they very often are capable of distinguishing between left-handed and right-handed molecules that they encounter out there in the real world. There is another example of stereoisomers in biology and medicine that I think is pretty interesting. And it relates to something that we talked about when we talked about acids and bases. I told you earlier in the course when we were talking about acids and bases that your stomach produces acid, and the acid is used to help digest food that you eat. And the way that your stomach is acidified is that there are cells that sit in the lining of your stomach, and many of those cells have very tiny molecular pumps. And these pumps pump H plus ions, or protons if you want to call them that. They pump H pluses into the interior of your stomach, and since the amount of H plus ions that you have in a particular area basically reflects how acidic it is, if you pump many H pluses into the interior of your stomach, you make it more acidic. So it's these little molecular pumps, and the pumps are actually much smaller than what I've drawn here, but it's those pumps that actually acidify your stomach. And I said that sometimes these pumps can work too well, and they can pump too much acid into your stomach. Some of the excess acid bubbles up into your esophagus, and it starts to irritate your esophagus, and it gives you heartburn. And one of the medicines that you can take to treat this type of heartburn is Prilosec. And again, many of you may have heard of Prilosec. It's an over-the-counter medication. And what the Prilosec molecules do is they basically, once you eat them, they break apart into individual molecules, and those molecules go to these pumps, and they jam up these pumps. And if they jam up the pumps are no longer able to throw H pluses or to pump H pluses into the interior of your stomach, and eventually some of this excess acid is sort of flushed away and your heartburn goes away. So that's how Prilosec works. If you look at the Prilosec molecule, it actually turns out to be two different molecules. Here's molecule number 1 on the top, here's molecule number 2 on the bottom. Again, hopefully you've come a long way and this doesn't look too intimidating anymore. These corners are supposed to be hydrogens. You can see some double bonds, you can see some single bonds, you can see a lot of different features of these molecules. You can even see this solid wedge here, which means that this particular region of the molecule is pointing toward us in 3 dimensions. These little 2 dots we haven't discussed, but they're supposed to represent 2 electrons attached to this sulfur atom, and those 2 electrons are pointing away from us because it has a dashed wedge here. But the Prilosec medicine actually has 2 molecules in it, this one on top and this one here on the bottom. And if you look at them, they are mirror images of each other. They are stereoisomers. In other words, they're not the same thing, they're just mirror images of each other. So when you ingest a Prilosec pill, you're ingesting these 2 molecules here. So Prilosec is a mixture of those stereoisomers. The interesting thing is that only the top molecule, it's only this stereoisomer, works to stop heartburn or acid reflux, because these pumps here are also come only either as a left-handed form or a right-handed form. And so the pumps are chiral too. And what that means is, again, you can think of this molecule up here as being something like a left hand, and this molecule down here as being something like a right hand. And the pumps in our bodies only come as left-handed gloves. Because of that, only the left-handed molecule can actually attach to the pump and jam it or shut it off. The right-handed molecule basically can try as much as it wants to fit into this particular glove, but it is never successfully able to do so. And so this bottom molecule here, when you ingest it in Prilosec, that mirror image is actually pretty much just junk. It doesn't do anything. The molecule that's doing all of the work is this particular molecule up here. So this may seem a little bit strange to you that both of those molecules are in the Prilosec medicine, but only one of them works. The top molecule has, and the 2 of these molecules combined is what you're basically ingesting when you take a Prilosec pill. The top molecule all by itself has its own commercial name. You can guess what that is. The top stereoisomer is actually called Nexium. So Nexium, when you ingest a Nexium pill, the pill pretty much consists only of this molecule, the molecule that can actually jam the pumps, and it doesn't have this molecule in it anymore. And if you ever read some of the marketing material for Nexium, what they will say sometimes is they'll say you only need half as much Nexium to treat your heartburn as you do for Prilosec. And the reason for that is pretty simple. It's because half of the Prilosec molecules that you ingest don't do anything. So if you have half as much of the top molecule as you do of both of those mixtures, it works just as well because all of the molecules that you're ingesting in Nexium are actually working toward jamming those pumps and relieving your heartburn. So that's an example of stereoisomers being used sort of to make medicines. Sometimes at this point the students stop me and they say, well, let's go back to Prilosec, and what's the point of making both of these molecules and putting them in the pill if only the top one works and the bottom one is junk, as you say? The reality is that when organic chemists make molecules like this, when they're trying to make medicines, it is very often difficult to make one mirror image without making the other mirror image. So you can imagine a chemist who's the first chemist who ever made Prilosec ended up making both of these molecules. And then they basically, through a lot of work, figure out that one of these molecules inhibits or jams up these pumps. Which one does it? I don't know. Let's just package the whole thing together and we can figure that out later. And that's basically what ends up happening, is that it's hard to make one mirror image without the other, so you end up making them both. You package the whole thing along and you ship it out as a medicine, and you figure out which stereoisomer is actually the one that works at some point later in time. Then you figure out a way to purify the good stereoisomer, and then you repackage that as a new medicine. And there are many examples of this. I want to come back to this bottom point in a minute, but I'll show you a couple of examples. These are medicines that you may have heard of. The medicines on the left are basically stereoisomer mixtures. They contain at least two stereoisomers, and usually only one of the stereoisomers is actually doing anything in your body. So Celexa is, I believe, an anti-anxiety medicine, and it is a mixture of two stereoisomers. Some people may have heard of Lexapro. Lexapro is basically contains only the purified stereoisomer of Celexa that actually does anything in the body. Same thing with Ritalin. Ritalin is a mixture of stereoisomers. There's another medicine called Focalin that contains only the active stereoisomer. Same thing, Prilosec and Nexium. You may have taken Ventolin for, I believe, some type of asthma, and there is a purified stereoisomer medication called Xopenex. So these types of stereoisomers show up a lot in medicine because many of our biological molecules also come only in one stereoisomer form, and so usually you need one particular stereoisomer of a medicine to work with our biological molecules. The other thing that I wanted to point out is that there's a special name that I'm just bringing up now. Molecules that have non-identical mirror images are sometimes called enantiomers. There's a lot of sort of sophisticated naming that goes into different types of enantiomers. We're not going to go into any detail here. All I want you to know is that if you have two mirror images, like is shown here, this is the top molecule is a mirror image of the bottom molecule. Sometimes these are called enantiomers of each other. And so that's just a word I want you to know. That means non-identical mirror images. As far as summarizing Chapter 12, this is what I want you to know about chirality or stereoisomers. The first thing I want you to be able to do is, if I show you a simple molecule, I show you a simple organic molecule, you should be able to tell me if it has a non-identical mirror image or not. And basically the test of this is, does it have a chiral carbon atom? If it has a chiral carbon atom, in other words, if you have a carbon atom that has 4 different things attached, then your molecule will have a non-identical mirror image. If it doesn't have any carbon atoms with 4 different things attached, then any mirror image that you draw will just be the same molecule that you started with. Second thing, you should just have a general sense of what chiral means. Chiral means that the molecule or object, it could be a glove as far as you're concerned, that the object has either a left-handed or right-handed feature. You should know what enantiomer means. Enantiomer is probably a little bit more sophisticated, but as far as we're concerned, what it means is a non-identical mirror image of another molecule. And you should know why enantiomers are important in medicine, and sort of be able to have an understanding of the example that I showed you a couple of slides back talking about Prilosec and the difference between Prilosec and Nexium. So that's it for Chapter 12. We're going to do much more detail with Chapter 13.